Hello, good morning, good afternoon, whatever it happens to be, wherever you're at as you're watching this. Welcome to Biochemistry. Biochemistry, Chapter 7 Carbohydrates and Glycoproteins. It's going to talk some fun stuff here. Now, carbohydrates. Carbohydrates are going to be what we refer to as aldehydes or ketones. Okay, aldehydes and ketones. What we're looking at are these carbon chains that have at least two hydroxyl groups or substances that yield a hydroxyl group during hydrolysis. Okay, remember hydrolysis means we're going to be cutting things up. So you're going to yield a carboxyl group. So it may be, a polymer of sugars, but when we go and we cut that glycosidic linkage, and we'll talk about what that is in a minute, between the sugars, when you go, the hot cutting, hydrolysis, you're going to be adding water back, a hydroxyl and an oxygen. Well, that hydroxyl is going to be bound to a carbon that already has a carbonyl group, so you end up with a carboxyl group. When you look at these carbohydrates, you're going to notice CH2O. That is going to be our base formula. And is how many carbons? Okay, glucose, 6 carbons. 6 carbons, 6 times 1, 6 times 2, 12 times 1, 6. C6H12O6. You've learned that since you were a kid. We split up the carbohydrates based on, well, how many molecules of sugars are there? Monosaccharides, saccharide meaning sugar. These are the simple sugars. A-glucose, A-fructose, or some form of 3-carbon sugar with the hydroxyls and whatnot. If you got a few of them joined together, a few, more than 2, we refer to that as an oligosaccharide. Short chains of monosaccharides. Okay, they're going to be joined by those linkages you learned back in intro cell and molecular, the glycosidic bond. Why did I say a few, 3 or more? Well, because we have a special designation if it's just 2. 2 bound together, di. We refer to that as disaccharides. 2. Way more than 3, well, we just refer to that as polysaccharide. So when you go and think about it, we have, 1, some, 2, or really a lot. When I say really a lot, I'm talking anywhere from hundreds to hundreds of millions. The carbohydrates can have multiple chiral carbons. The configuration of the groups around each carbon atom determines how that compound interacts with others. Yeah, kind of like how we saw with the amino acids. Okay, the central carbon, the alpha carbon, is the chiral center, and then everything attached to it can rotate around that. Same here with the sugars. Okay, but here, instead of an individual carbon, it's going to be the actual glycosidic linkage we're going to see, and where it's a chiral, and anything attached to the sugars, we're going to have chiral centers there, which is why you end up with, D, L, and D. Just like we saw L and D, depending upon which way everything's turned in the amino acids, we see the same thing here with the sugars. Our monomers, our building blocks, are going to be referred to as monosaccharides, okay? I used the stupid analogy in freshman bio, and it still holds true. Instead of thinking monomer subunits, think Lego bricks. Pretty much every kid, even adults, have played with Lego bricks of some sort. Well, individual bricks are the monomers. You build them up, well, whatever you built, that house, that wall, that plane, that spaceship, that's the polymer. It's made up of many, many, many subunits, individual Legos. Why do we do this? Well, it turns out that individual monomers, if you have 1,000 sugars in the cytoplasm of a cell, actually affects the subcellular environment greater than if you linked them all together as a polymer. So as we see, the polymer form avoids high osmolarity. Okay? Inside the cell, that one polymer, well, it's 1 in, 0 out, as opposed to million in, 0 out, and then water has to try to balance. We see that has a less of an impact on osmolarity or, also diffusion. We study the sequence of these, okay? The sequence of the polysaccharides, orientation of the molecules within the sugars will affect the overall three-dimensional structure, just like we got done talking about with protein chemistry. The sequence of amino acids will impact secondary structure. Secondary structure impacts tertiary. Tertiary impacts quaternary. We're going to see the same thing here. Because polysaccharides are not just a straight chain of sugars. Okay? The sugars and whatever's attached to the sugars, what microenvironment within the cell or within the tissues those sugars happen to be in when they're in the polysaccharide form will cause them to twist and turn, form, loose helical spiral structures, will form branching structures. And that's what we're going to be looking at this chapter. So, we're also going to see why structure dictates function with these. Certain three-dimensional structures that these polysaccharides can take will have functions within the cell. Certain, three-dimensional structures will impact whether or not other, macromolecules, specifically enzymes, can interact with them. And there's, just like with the proteins, there's an infinite variety of how we can link these together. What's the orientation of the glycosidic bond? Is it above or below the plane? Are we going to see a chiral, turn, which then will lead to, some loosely helical pattern? Are we going to be seeing branching? To what degree of branching? How much branching? Things like that.